Okay, good morning. Welcome all to today's lecture. And as Jeremy said, today we will be focusing on modeling genotype by environment interaction. So the contents of the first lecture are that we quickly go over different types of environments and also the size of observed the genotype by environment interaction in animal breeding. We'll spend some time on how to estimate G by E, and then, if time permits, we will go over some different data structures needed to estimate G by E. So learning outcomes of this part are mostly that you're able to understand, but also able to apply some of the basic concepts related to G by E, and that you also understand the different terminology, because throughout the literature, there's a number of different terms are used to denote the same thing. And also that you understand the reaction norm model and how to translate results of that model into outcomes specific to certain environments. So to start slowly, some basic definitions. I'm sure you're all well aware what phenotypes, genotypes are, and environments, and then the interaction between them. That's what we are concerned with in this first lecture. So if we talk about reaction norms, this typically refers to, let's say, the response of an individual, the phenotype of an individual or a genotype to a particular environment that is expressed on a continuous scale. So here we have two different individuals, and you can see that these individuals, if the environment along the x-axis changes, their phenotype respond differently to changes in the environment. So if this is happening, if we see this differential response to differences in the environment, then we say that there's genotype by environment interaction. And one critical thing here is how do we define this environmental gradient? So another term that's commonly used in the literature is environmental sensitivity. And this refers to the slope of the reaction norm. So if the reaction norm is flat, basically the individual does not show any sensitivity to changes in the environment. But there's a different term that's also closely related to this called phenotypic plasticity. So we could say that the slope of the reaction norm also measures the degree of plasticity of the individual. So just to show this in a graph, the individuals with a flat reaction norm, they're not sensitive, and the ones that have a steeper gradient are increasingly more sensitive. So just to compare three different possible outcomes if you try to do this exercise. First of all, it's possible that you see there's no genotype by environment interaction. That does not mean that the phenotype does not change with changes in the environment. It simply means that all genotypes or all individuals respond in the same fashion. So in other words, the reaction norms of the different individuals, they are parallel to each other. Another possibility that may happen is that the reaction norms are not parallel, but they're also not crossing over. So this means that in this case, in any environment, the green individual has a higher phenotype than the red one. But the relative difference in phenotypes between the individuals does change with changes in the environment. And then finally, the one that we are, as animal breeders, are typically most concerned with is if there's a re-ranking, and this happens as soon as the reaction norms they cross each other. So in this case, in one environment, the red individual would be have the best phenotype, and in another one, the green one would have the best phenotype. And we estimate this. If we talk about the scaling, we look at the homogeneity or the heterogeneity of variances across environments. If we look at the re-ranking, then we measure this with the genetic correlation between environments. So in the top two cases, this genetic correlation would always be 1, because the ranking of the individuals is always the same. But in the the bottom right figure there, the correlation is lower than 1 between the extreme environments. So let's have a look at types of environments, but also G/E that has been found in animals. We can define environments at very many different scales, and perhaps, well, if you look globally, I guess you are well aware that close to the equator, the circumstance and the climate is very different than it is perhaps up north in Scandinavia or here in northwestern Europe. So that would be a very a big change in environment. But environments can be defined in very many different ways. So next to the climate, it can be the housing system, like are animals kept indoors or are they kept outdoors? The nutrition of the animal, disease pressure, but also stocking density. Is there a lot of individuals in a small amount of space, or do they have a lot of space to freely roam around? And there's probably a bunch of others that you may be able to think of. So if we try to structure this a little bit, we could say that there's the environments we could call the most large differences. We could call this differences in mega environments, in macro environments, or micro environments. And like the more precise you define certain circumstances, for instance, in micro environments, this could be within a country to look at differences between herds. And then, well, the smaller the differences in environment, the smaller the magnitude of the G/E that you may find. Another way to categorize or to group types of environments is to look at those that are categorical. So if you have different farm types or system types, or the presence or absence of a disease, then you can very clearly define individuals in 2 or more different groups. But there's also environmental measures that are more continuous. So if we think about temperature, day length, or rainfall. So if it's categorical, we typically would try to model G/E by bivariate if there's 2 environments, or a multivariate model to estimate the genetic correlations between all those pairwise combinations of different discrete environments. And if it's continuous, then probably the reactional model is the better model, because that can really take into consideration the continuity of your environmental descriptor. So how do we quantify the size of the G/E? I said it already that the most important parameter to look at is the genetic correlation between 2 different environments. And that's already coined like something like 70 years ago, that this is the way to look at this by a falconer. And then if that's the parameter to look at, then the question really is, how much does this genetic correlation, does it deviate from 1? And sort of a rule of thumb that people have come up with, if it's 0.85 or higher, then maybe the differences between environments are relatively small and you should not worry too much about this in a breeding program. But if they're below this value, it's probably good to think about how are you going to deal with this. So I'll just quickly run you through the different livestock species and say a little bit about genetic correlations between environments that people have found in different studies. So in dairy cattle, and a lot of studies have been done there. This is also partly done by an organization called Interbull that tries to estimate breeding values using information of different countries. And to do this, they have this model that actually considers each country to be a different type of environment. So they have estimated genetic correlations between all pairs of countries involved. What we see there is that for production, typically correlations are higher than 0.8, and the more closely related the systems in the different countries are, the closer these values are to 1. So you can think about the lowest values of 0.8. You may see this between let's say, large-scale facilities in the US, where all the animals are kept indoor versus grazing systems such as in Ireland and New Zealand. If you look at fertility and longevity, then there's a larger bandwidth of correlations, and some are as low as 0.5, and then it really starts to have an impact. Then we in our group, we did 2 reviews, one in pigs for this for genetic correlations between different environments, and both for pigs and poultry, the typical contrast to look at is on the one hand is the environment that purebreds in a breeding program that they experience. So the breeding stock is very valuable for breeding companies, so they are kept in high biosecurity systems where they're very much supported. Whereas these individuals, the products that are derived from them, they are kept under commercial circumstances where there may be higher disease pressure and a lower level of management. So there's a lot of studies that look at the correlation between those systems where the purebreds, the selection candidates are kept, and the crossbreds, which are the commercial end product. So when we reviewed this correlation across 27 studies on pigs, we found an average estimate of 0.63, and this correlation was rather similar across different trait categories. We did a similar exercise for poultry. We included both laying hens and broilers, and there the number of studies was even fewer, where we just found 19 studies in total. But then, based on these studies, we made this figure where we group traits that are more or less related. So there's a number of egg-related traits, then there's body weight and conformation-related traits on the right, and in the middle there's a group of other traits. And what you can see is that generally for the egg number traits, the correlation are relatively high. For the body weight, it tended to be somewhat lower. There was a very low estimate for body conformation, but there was very little information for it. And then in the middle, we see that for mortality, it was actually also not too bad. But then there's also one here, and that's actually, those values come from a study by Nicolas Bedet, who's here with us, where the authors looked at the genetic correlation between resilience, and I believe it was the LM-VAR parameter that you looked at, between purebreds and crossbred, and this was actually reasonably low with a value of 0.4. And I think this makes quite a lot of sense because, as I said, the purebreds, they are in a very supportive environment, and the crossbreds, they're in a much more challenging environment. And that's probably also going to not only affect the expression of the resilience of individuals, but it may also affect the nature of the resilience or the environmental perturbations per se, such that it really is a different trait in those different environments. And then finally, in aquaculture, there's been an extensive review. The thing with aquaculture is there's many different species, also quite a number of different traits, and there's a large variation in this genetic correlation has been observed. And probably because to some extent there's such a variation in systems, some are very well controlled, other ones are just cages outdoor in the sea that are perhaps much less controlled with a lot of disease pressure. So it makes sense that we see a lot of variation in this genetic correlation there. Okay, let's move on then to statistical methods to estimate G by E. If you have any questions in the meantime, just feel free to ask. Yes.
When you estimate all these experiments between purebred and crossbred animals, the part of the G by E is also G by G, isn't it?
Yes. So that's a very good point, and we also pointed this out in one of those 2 papers I just showed, is that the purebreds and the crossbreds, they are partly different genotypes, right? And the crossbreds, they their performance may be affected by non-additive effects because they are crossbreds, such as heterosis, and dominance is more likely to play a larger role in the crossbreds than the purebreds. So the thing is to discriminate between this, this is you need a 2x2 design, right? So we have one genotype is in one environment and the other one is in the other. So you would need to fill one of the two other cells at least to be able to discriminate between those two. And we tried to have a little bit of a go at this in the review paper, but it was not very straightforward. But you're very right that these correlations that we see between purebreds and crossbreds, their deviation from one cannot be only explained by G by E, but there's also genotype by genotype interaction, if you like.
You need to have a dominance to have a very strong.
I'm not sure if that's particularly needed. No. And I think we sort of more or less concluded that most of this magnitude of this correlation that we see is due to genotype by environment interaction. So, yeah.
Yeah, I also have a question.
Yes.
So I as a person never really understood the concept of genotype by environmental interaction in animals. I mean, in plants, it's very easy to have same genotype, like in different countries, different short. But in animal, we can have particular animal.
Right.
Several places, so it's always like.
I'll try to answer that question throughout the lecture, somewhere towards the end, and if I didn't, let me know. Right? Then yeah. So the methods that we use, the statistical methods to estimate G by E in animal populations, we use pedigree relationships between individuals, and we typically use blood to estimate breeding values. And the reason is that our main interest is in breeding values or additive genetic effects. So then the most common models to analyze G by E, once again, are bivariate or multivariate models or reaction norm models that are implemented using random regression. So both of them require that we determine variances, the pattern of the variance across different environments, but also the covariance between them. We need those two different components to estimate genetic correlations between environments. So with the multi-trait model, we discriminate discrete environments, for instance, grazing and non-grazing, and then we estimate the genetic correlation between those systems. With the reaction norm model, we measure the environment in a continuous way, and this could be something like herd average performance, but it could also be the temperature humidity index. So the multi-trait model, what does it look like? Basically, what we do for every environment, we construct a sort of a model, separate model statement that states that the phenotype of a particular individual is equal to its breeding value, a bunch of fixed effect, and the residuals. And this breeding value then pertains to that particular environment. So if we would have two environments, we have two model statements, and we get two breeding values for each individual in the system. So then we get estimated breeding values for each individual in each environment. We also get genetic variances for each environment and the correlation between those. So basically, if the number of environments is n, we get an n by n genetic variance covariance matrix. For the reaction norm models, it looks a little bit different. We have one model statement that captures all the different environments and possibly repeated observations of the same genotypes. So we have that the phenotype is equal to a bunch of fixed effects. Then we have this Bx here. That's actually typically a population average curve from which we model the deviation of the individuals to be their breeding values. And then in the simplest case, if we have a linear reaction norm, the individuals have a breeding value for what we call the intercept. So that's a constant breeding value term regardless of the environment, plus 1 for a slope. And this is obtained by doing a so-called random regression of the phenotype of the individual of an environmental descriptor. So if we do that, we get a 2 by 2 for this linear reaction, and we get a 2 by 2 variance covariance matrix. We get a genetic variance for the intercept and 1 for the slope, and the covariance between the two. What we get with the multi-trait model is that for every environment we have a genetic variance, a separate genetic variance, but we also have a separate residual variance. So if there's any heterogeneity of residual variance across environments, the multi-trait model sort of naturally takes this into consideration. What we do in the reactional model is typically we would make groups of different environments that have environmental descriptor values close to each other, and then model a separate environmental variance for each of those groups. And the number of groups to consider would be typically anywhere between, say, 3 to 10, depending on the amount of data, but also the difference in this environmental descriptor values. And then the reaction norms can take any shape. So this is a copy of my PhD thesis. I spent 4 years of my life running this kind of models. And here we did, I think this was a second order, so it wasn't a linear, but it also had a quadratic term. And then the reaction norms are not strictly linear, but they can take possibly any shape. And that flexibility is nice, but you have to be careful and actually look at what is the outcome of your model to see if things make sense or not. So this, yes. Yeah, that's a fair question. I think you would probably do it by number of individuals. Such that if this environmental descriptor is normally distributed in the middle of the environment, there's probably a lower bandwidth than in the more extreme cases. Yes. Yeah. Or you could try to make it sort of bins of equal width in terms of the environmental descriptor can make it this, but you should be careful that especially at the extremes, there's enough individuals in there. Yes. So there's a number of issues with reaction norms, which covariate to use. Should it be something that you measure at the environment directly, or is it something that we can derive from the data? And I'll show you this later. You have to think about scaling of the variance and stuff like this, and also whether to use polynomials. Then, which I won't be going into, is that there's something to do with the model comparison. So the idea is that you start a model with only an intercept. So there's basically a model that doesn't include G by E. Then you add the linear reaction norm, and then you can add a quadratic component or a cubic, etc. But every next step, you compare the two nested models to each other to see if the next one actually improves the fit to the data compared to the previous ones. And then finally, something about interpretation of results. So which covariate to use? Well, you can try and measure anything in the environment, like obvious things such as temperature, daylight. If you think about fish, it may be the oxygen level in the water or the salinity level. But then the question really is, well, how many things can you measure? Do you want to test all of these? And a pragmatic solution to this is to use what we would call internal derived factors. And they're basically for a group of individuals that are in the same herd or the same contemporary group, that we use the mean performance of these individuals. And the idea is that the mean performance of these individuals is a reflection of the environment as they have experienced it. And in many cases, this is also what I did in my PhD thesis, we would first run a model where we include a herd year season effect, and then the estimated effect for the 30-year season is then the environmental descriptor and the reaction normal that you would run subsequently. So if we think about temperature, and more about this also in the next lecture at 10 o'clock. But temperature may affect phenotypes only above a certain threshold, and this has been recognized for quite some time. Here's an example in pigs where we see the trait varying rate at first insemination. These are average values for 2 different breeding lines. And what you can see is that at the lower maximum temperature at the day of insemination, the one line actually outperforms the other one. But the other one that has a lower performance keeps this performance also at higher temperatures. Whereas the higher performing line past a temperature of about 18 degrees, there the phenotype starts to go down. So then if you wonder like which breeding line to use, then it really depends on your system or your production system. Do you mainly experience lower temperatures, then you might go for the higher performing one. But if you're in a certain part of Europe, maybe you want to go for the other one. If you target anything you measure in the environment to discriminate the environment, it's good to think about the nature of that particular parameter. And also the distribution of the values that you can expect. So if you would look at day length, it's good to realize that that this follows a sinus function throughout the year, so that if you plot all these environmental descriptors, maybe you will not see a perfect normal distribution, but that's probably also because you shouldn't expect it. So then, as I said, the pragmatic way to go about this is to use 30-year season estimates, typically of the same trait that you're modeling the G/E for. And the strategy used to this is that you use the same data, the same mixed model, but then only there's no reaction norm in it yet. You first estimate these 30-year season effects, and then in the next model, you use the estimates of those 30-year seasons as the environmental descriptor. You do use the data twice, and that can be a little bit tricky. So, and one issue with this is that the performance of the individual itself also affects the estimate of this 30-year season effect. So if the individual is very good, the performance in the environment may also be a little bit higher, and this also may sort of be a self-fulfilling prophecy across these 2 different models. So possible solutions to go about is that you derive the environmental parameters actually from a different trait that's perhaps not closely related to the trait you're modeling. You could use another group of animals to calculate the environmental parameters, but this may often not be possible because you would need a large number of data to do this. And well, the pragmatic way to go about this is, as long as your 30-year season groups have sufficiently large number of individuals, the impact of the phenotype of the individual itself on that 30-year season estimate should be relatively small, and you might be quite okay in following this approach. In my PhD, I did look a little bit into this, and what I found is that with reaction norm models, we typically, amongst others, due to this thing I just explained, we tend to underestimate G/E. So here I did some simulations, and in this graph, what you see is the solid black line is the genetic variance, the simulated genetic variance across environments, and you see that it's low in the, let's say, the lowest environment, and it's very high in the most supportive environment. And then if I re-estimate those variants with reaction norm models with different settings, in all cases, the difference in the variance between the environments was smaller than what I had simulated. So based on this, we concluded, and also based on the estimated correlation compared to the true ones, that with these models, we tend to underestimate the degree of G by E. One possible solution that has been put forward almost 20 years ago is to actually to build a model that estimates the Hertzian effects and estimates the reaction norm at the same time. So there was a group in Denmark that worked on this, and they built this Bayesian models that can do it all sort of simultaneously, and they should try to mitigate some of these issues. But the modeling is complex, and the implementation of the model also is. So although this was quite a neat solution, this hasn't really found uptake afterwards. Then let's talk a little bit about the scaling of the effects and predominantly the environmental descriptors, but also use of polynomials. So first the question is, yeah, do we need those polynomials? So polynomials, they are, the way it works is that if you have this linear reaction norm model, basically you do a random regression for each individual. For the intercept, you do a regression on a value of 1 for all individuals, right? And then for the linear one, we have a value x that may be different for each individual depending on the environment where they're in. Then if we go to the quadratic scale, we get a value that's proportional to x squared. As soon as we go to the quadratic scale, the issue that we see is that there's going to be a relationship between x and x squared. These two are not independent from each other. And the polynomials, what they do is they try to transform those. If we have a quadratic reaction norm, so we have 3 covariates, they transform those covariates such that they are more or less independent from each other. So if we do a linear reaction norm, there's no need to use these polynomials because, well, there's no variation in the covariates for the intercept, so there's not really a relationship between the covariate for the intercept and the slope, really. So you can do it, but it doesn't really make a difference. For higher order reaction norms, such as we would call the quadratic one, we would call this a second order, and then if we have something to the third, we would call this a third order, etc. Then it becomes important because it will make these covariates more or less independent, and this will really improve the properties of solving the model. So the more related those covariates are amongst each other, it also means there's more relationship between the breeding values for these different components, and then it becomes quite difficult for the model to properly solve this. So if you run a model like this in a package like ASRAML, that would have standard options to compute these Legendre polynomials. So another word of saying that they're more or less independent is that they are orthogonal. And the typical approach that's taken is that you first scale your environmental descriptors to be somewhere on a scale between -1 and 1. And then if you go to the second or higher order, you use these Legendre polynomials that are estimated following sort of a fixed function. Then a little bit about the interpretation of results, particularly of reaction norm models. So what we get with the multivariate model is a genetic covariance matrix between different environments, and that's ultimately also what we are interested in. But we don't get this straight away from the reaction norm model. So if we want to compute this covariance matrix between different environments, let's call this matrix H. We can obtain this by taking the matrix between the estimated covariance for the different orders of the polynomial. So if we have a linear regression norm, this would be a 2x2 matrix. We have a variance for intercept and one for slope. And we would pre and post multiply this with a matrix of values of the orders of the polynomials for the environments of interest. So if this matrix P, it could actually look as follows. If we have a number of different environments where we simply have different values of the environmental descriptor, we build a matrix with ones and with all these different environmental descriptors, or the polynomial derivation of those, and we remultiply the matrix G with this matrix, and we post multiply it with its transpose. Then, yes. It would have the n here, the number of lines would be the number of environments for which you want to get the variances and the covariance between all those environments. Yes. So you're fully in control of how many those are going to be, right? You can make this very large or very small if you want. But the idea is that these values of x, they cover the entire range of different environmental descriptive values that you may have seen. The one thing is that, especially in the extremes, you do not want the lowest value of the environment that you've seen. You don't want to include this in here, because that's only individuals that were in that herd environment have experienced that particular value. So there's very little data there. You want to probably be a bit closer to the averages to report extreme observations. So then, if you have this full variance covariance matrix, one of the things you can do is actually compute the correlation, genetic correlation between different environments. So here there's a study where on the x-axis there's challenge load for a particular disease or health status. So the idea is if the load is lower, there's a higher health status, right? So we see a blue curve here that's indicated high health. The correlation that's on the y-axis for this curve starts at 1. So this means that the high health environment was actually defined to have a challenge load of about 7.5. So that environment with itself has correlation of 1. But then the blue curve indicates, okay, what is the genetic correlation between this particular environment and an environment that has a continuously higher challenge load? And you see that if the difference between those environments becomes bigger, the genetic correlation becomes lower, to the point where between the extreme environments, it's about 0.5, 0.4, 0.5. So we can do this taking as one environment, the one with the highest health status or with the lowest health status, and then we get this other curve. And then we sort of see the opposite pattern. And then finally, the one in the middle takes as a starting point the average or the intermediate environment. So we see a genetic correlation here of 1, because that's with the environment itself, and then to both sides it starts to drop off. Okay, then let's talk a little bit about data structures to estimate G by E, and that's going to answer one of the questions that was posed earlier, like how can we estimate G by E in animals if we're not able to put the exact same genotype in different environments. So if we want to estimate G by E and we have categorical environments, then indeed the ideal design would be to have the individual itself to observe its phenotype in those different environments, right? Because then you see the same genotype in the different environments. That's perhaps still not ideal. It would be even better if you had clones of that same individual and put those in the different environments. But yeah, we simply don't have them, or, well, we're not allowed to create them for whatever reasons. So often the other restriction is that animals perform in only one environment. Moving animals from one environment to the other can be a hassle for whatever practical reasons or disease restrictions, stuff like this. So because we're typically not able to do this animal breeding, what we use is quite extensive databases with animal phenotypes and the pedigree of these individuals. So we see two animals that perform in different environments, but we know what is the relationship between them. And based on this relationship, we can see, okay, the degree to which these individuals are the same. How does that explain the difference in performance between the two different ones? So they're not the exact same genotypes, but they're partly the same genotypes, and we can leverage that link, that genetic relationship between the individuals to capture the G by E. That's one way to do it, to use pedigree to create the links between individuals in different environments. Typically now we increasingly more use SNP genotypes to do this. So traditionally, pedigree links are used, and a very common approach is to use half siblings or half sisters in different environments. So then what you do is you have a bunch of sires, and you have two different environments. You make sure you get offspring of those same sires in the two different environments. Why do we focus on sires? Simply because with sires, with semen, it's easier to make a large number of offspring of the same individual. And then the stronger the relationships between the individuals in the two environments, the better the information is to estimate the G by E. So if we have half siblings, the relationships between those is 1/4, right? And that's in many cases probably the best thing we can do. Yeah. But it may be that we try to estimate the degree of G by E based on less related individuals. It's possible, but then we simply need more data, because if the connection between the individuals is smaller, we just need more data to be able to estimate something properly. And one thing that helps is to use genomic relationships, simply because they are much more precise than pedigree-based relationships. So if you would want to do an experiment like this, it's a good idea to start thinking about it beforehand. How big should the experiment be to be able to get an estimate that's actually useful? So in other words, useful, you could translate this in that the standard error of the estimated genetic correlation that you're going to obtain should be reasonably low. Because let's say if you get an estimate of 0.6 for the standard error, for the genetic correlation, but if the standard error is 0.4, well, you don't really know too much about what the genetic correlation really is, right? So in the traditional modeling this, then what we would evaluate is the number of different families. So if we think about sires having offspring in different environments, then the number of families is basically equal to the number of sires used in the experiment. The number of offspring for each of those sires in the different environments, and then also the heritability. So the higher the heritability of the trait, the smaller the design can be to get the same a certain level of standard error. So there's a number of formulas in the literature that allow you to predict those standard error. They're typically a function of these parameters that I just mentioned, like number of families, size of them, but also the heritability. And here, this one is based on the intra-class correlation between the different groups. And that's sort of, let's say, the correlation between the different families. So you can work out that intra class correlation between half-set families should be equal to a quarter times the heritability. Two of our colleagues developed this equation to predict the standard error of the genetic correlation. And in that case, the idea was if we have purebred and crossbred environments. So we again would have sires that have both purebred and crossbred offspring. And well, it looks a little bit nasty, the formula, but if you look at it a little bit, one thing that comes back in all these formulas is the genetic correlation, the value of the genetic correlation itself. And here, rather than having the size of the families, there's terms that actually predict the reliability of the breeding values of a sire in a particular environment. And those in themselves, they are functional. The heritability of the trait, but also the number of offspring. And actually, on one of the review pages that I showed you earlier, we validate this formula against reported standard errors by different studies, and we found that this formula works actually pretty well. So the nice thing with these formulas is that once you have them and you've established them, you can use them to do some power calculations, right? You can just play around with the parameters that go into the formula and then see what you get out. So here, for a particular situation, if we assume a genetic correlation between environments of 0.7 and a heritability of 0.3, then as function of different family sizes, the obtained standard error or the expected standard error is plotted, given that between the different environments, the degree of relationship is those between the grand offspring of sires, half-step offsprings, or clones. And what you can see is that actually, whether you have clones or half-step offspring, the difference between it is relatively small in terms of the standard error as soon as the families are sufficiently large in size, so let's say 50 or more. But if you have very few individuals per family, there would be a benefit of having clones if you would be able to create them and actually would be allowed to use them. Conversely, if the relationship between the individuals is that to the degree of grand offspring, then even for larger family sizes, the standard errors can be quite a bit higher than if you would have relationships for half-step offspring.
But that is provided you have a given number of families.
Yes, yes. Yeah, so there's 100 families in all these cases here. Yes.
But with the, I think I have used the Robert's formula. I mean, what the output is that you get, I mean, you optimize the number of families and the size of the families at the same time.
Right.
No?
Yeah, so if you think in terms of an experiment or testing capacity, maybe you can have 5,000 individuals in one environment and 5,000 in the other, right? So then you have to indeed optimize what is the number of sires and thus the family size, right? Yeah, yeah. So if you have too few sires, that will not be good. If you have too many, each of them has very few offspring, so that the answer is probably going to be somewhere in the middle. Yes. And then with in the last 10, 15 years, with genomic data being increasingly more used, there's also formulas developed that try to predict the standard error of genetic correlation if you use genomic information. And here the new term that sort of enters in this equation related to genomic information is this ME, which is also termed the effective number of chromosome segments. And that can be computed as one of the variants of difference between genomic and pedigree-based relationships. So the idea is if there's a lot of variation in relationships, then this ME is actually relatively small. So more variation would be useful here. If we take that formula into consideration, then we can again do sort of the same kind of experiment. work through these different formulas. Here we have the number of animals per environment. Again, on the y-axis, the standard error of the genetic correlation. And then what we can see is that if we have pedigree half-zips or genomic information, that's all, that all yields relatively similar standard errors of the genetic correlation. Unless the number of animals per environment becomes quite low. So just to summarize, if you want to estimate genetic correlations between environments, you need typically large data sets, at least to be able to accurately estimate those genetic correlations. Something like at least 50 to 100 families, each with a 50 to 100 offspring. Clones may be better theoretically than half-zips, but they're actually, if the family sizes are substantially large, then half-zips are quite okay. And grand offspring is quite a bit worse than half-zips. And finally, with genomic data, we can decrease the standard errors quite a bit, especially with limited experiments. So if there's fewer than a few thousand individuals per environment. Then to wrap up the part on estimating the G/E, we can either use multivariate models if the environments are discrete, or reaction norms if the environments are more continuous. Reaction norm models are a bit more complex. You have to think more about how to translate the outcome to what you're actually interested in, but you also have to be critical to the estimates that you get. And different environmental parameters can be used in reaction norms. Typically, we use herd season estimates, but you have to be careful with this approach as well. So that's what I had for this part. Maybe you want to take a short break before we go to the next one.